are fine. So in this video we will discuss about the organelle of the cell. Okay, in last video we have already discussed about membrane, about nucleus, okay, plasma membrane, and also the cell wall. Okay, so in this video we will discuss about the organelle in the cytoplasm. Okay, in this video we will discuss about the organelles, okay, which present in the cell wall. So let's start. Here every cell has a membrane around to keep its own contents separate from the external environment. It means every cell have the membrane. Which type of membrane present is called plasma membrane. Okay, in this, and this is an animal cell, so this layer is called plasma membrane. Okay, and to protect the cell that any material cannot go out and not come inside the cell. Okay, separate. Okay, it separate the cell. Large and complex cells, including cells from a multicellular organism, need a lot of chemical activities to support their complicated structure and function. It means in the multicellular organism, there is a large of function activities are performed. For this, we have to support the complicated structure and function. Okay, to keep this activity of the different kinds separate from each other. Okay, to keep this activity is separated from each other. These cells use membrane bounded like structure or organelle within themselves. Okay, this type of cell use the organelle or a membrane bound structure. This is one of the feature of the eukaryotic cell that distinguishes them from prokaryotic cells. Some of these organelles are visible only with an electron microscope. Okay, in the cell is also only visible, also can be visible by microscope. And we if we want to see inside what is inside the cell, then we have to see with the electron microscope, which is more powerful than the normal microscope. Okay. We have talked about the nucleus in previous section. Okay, some important example of cell organelle which we will discuss now are endoplasmic reticulum, Golgi apparatus, lysosome, mitochondria, plastid. They are important because they carry some very circular function in cell. Okay, these organelle are important because these organelle play a very major role in the cell. Okay, now we have first one is endoplasmic reticulum. Okay, so let's see about what is the function of endoplasmic reticulum. Endoplasmic reticulum, that is ER, is a large network of membrane bound tubes and sheets. Okay, it is a large membrane bound of what? Tube or sheet. Okay, it looks like a long tubule or a round oblong bags. Okay, it also looks like a tubule and like a bag. Okay, the ER membrane is similar in structure to the plasma membrane. Okay, it is a similar like a plasma membrane. Okay, not same but it's similar. Okay, similar. Okay, there are two types of ER: rough endoplasmic reticulum and smooth endoplasmic reticulum. Okay, here endoplasmic reticulum is divided into two parts, that is smooth ER and rough ER. Okay, so in the smooth ER there is no ribosome is present, but in the rough ER, it means rough endoplasmic reticulum, there is on the membrane there is a ribosome. Okay, you know what is ribosome? The protein synthesis. Okay, so the rough endoplasmic reticulum carries ribosome, that's why it is rough. Okay, and now here looks like a rough under microscope because it particles called ribosome. Okay, it has the particle called ribosome attached to its surface. The ribosome, which are the present in all the active cell, are the site of protein manufacture. I have already out. Okay. The manufactured protein are then sent to the various places in the cell depending on the need using the ER. Okay. It means the protein, if the if the cell if the ER synthesize the protein, then it sent to the various places in the cell depending on the need. Okay. The SER helps in the manufacture of fat molecule or a lipid, important for a cell function. Okay. Some of these protein lipids help in the building of cell membrane. This process is known as membrane biogenesis. Okay. If the protein is formed for the membrane building, then the process is called membrane biogenesis. Some other proteins and lipids function as an enzyme and hormones. Although AR varies greatly in appearance in the different cell, it also form a network system. Okay. So this is all about the ER, which is divided into two parts, smooth and rough. Okay. This is our cell. It means the eukaryotic cell, which have the nuclear envelope, mitochondria. And here we can see this one tubule-like structure is called ER, and this one is smooth. It means that there is no ribosome present. That's why it is called smooth. And the other one is you can see here ribosomes are present. Okay, so this called rough here. Okay, and here this one is called Golgi apparatus. So next, thus one of thus one function of the ER is to serve as a channel for the transport of material between various regions of the cytoplasm, between the cytoplasm and the nucleus. The ER also function as a cytoplasmic framework, providing a surface. Overall, it helps in protein synthesis, okay, and to transport also, okay. For some biochemical activities of the cell, in the liver cell group of animals called vertebrates, SER play a crucial role in the detoxifying many poisons and drugs. Okay, SER also helps to detoxify the poisons and the drugs. Okay, to destroy. Okay. Again, this is a plant cell. Okay. Here this is a figure of animal cell, and this is the figure of plant cell. Here we can here we can see here that this one is outer layer is what, cell wall, inner is plasma membrane, here cell wall, this layer is cell wall, and this one is, this black line is called plasma membrane, and this one is what, middle not, this is a space that the which name is called, yes, plasmodesmata. Okay. Now here cytoplasm, chloroplast, this one is chloroplast. Okay, this big structure is one vacuole, although this one is nucleus, mitochondria, chloroplast. Okay. So this is all about the figure of, this is all about the image of plant cell. And now we will read about the Golgi apparatus. It is also helps into same function like protein. Okay, synthesize the it is synthesized by the packed of ER. Okay, we can see here that you all can see here that this one is ER, and after ER the Golgi body is formed. So with the help of ER the Golgi body is formed. So let's read about this. The Golgi apparatus first discovered by the Camillo Golgi. Okay, it is who discovered the Golgi apparatus? Camillo Golgi. That's why it is called Golgi apparatus. This one. So consists of a system of membrane bounded vesicles, flattened sacs, arranged approximately parallel to each other in stalks, called cisterns. Okay, we can see in the figure that it runs like this. Okay, like a pattern. Sorry. These membranes, this is called cisterns. Okay, often have a connection with the membranes of ER. Okay, I have already told that it is connected with ER and therefore constitute another portion of a complex cellular membrane system. The material synthesis near the ER is packed and passed various targets inside and outside the cell through Golgi apparatus. The function includes the storage of modification and packaging. It helps to package and modification of vesicle. In some cases, complex sugar may be made. Simple sugar is in the Golgi apparatus. The Golgi apparatus is also involved in the formation of lysosome. Okay. See here. This is a nucleus. Okay. This one is nucleolus, and with the help of nucleus outer membrane, the ER is formed. Okay, and then after, with the help of this ER, what is this? This one is Golgi apparatus, and now lysosome. Okay, so this is the cycle. Okay. Here is about the camellia Golgi. And now next one is lysosome. We have to read about the lysosome and then mitochondria. Okay. So let's see what is about the camellia Golgi. Camellia Golgi was born at Corteno near Brescia in 1843. He studied medicine at the University of Pavia. After graduating in 1865, he continued to work in Pavia at the hospital of St. Matthew. And that time, most of his investigations were concerned with the nervous system in 1872. Okay, it means Camellia Golgi was born at the Corteno. This one is a place of that where the Camellia Golgi is born. Okay, and now next one is in which year? In 1843, and then this one is the all about the education system that he qualified. Okay, and what he done before graduating. Sorry, after graduating, he accepted the post of chief medical officer at the hospital of chronically sick at the Abitibrescio. He first started his investigation to the nervous system in the little kitchen of his hospital. Okay, which he had converted into a laboratory. Okay, where he started his first investigation was on nervous system. Okay, in a little kitchen in the in this hospital. Okay, in the hospital there is a little kitchen. Okay, and in that kitchen he converted, the kitchen is converted into laboratory, and however the work of greatest importance which Golgi carried out the revolutionary method of staining individual nerve and cell structure. This method is referred to as the black reaction. This method uses the weak solution of silver nitrate is particularly valuable in tracing the process and most delicate. Ramification of cell all through his life is continued to work these lines. Modifying and improving this technique, Golgi received the highest honors and awards in recognition of his work. He shared the Nobel Prize in 1906 with Santiago Ramon Cazal for their work on the structure of the nervous system. Overall, he worked on the nervous system. Okay, and he also get the information about the nervous system. Okay, so for his great investigation, he also awarded with many prizes like Nobel Prize. Okay, so this is all about the biology. Now our lysosome. Okay, in this video we will only read about the lysosome, and again in next video we will finish the chapter. Okay, so structurally lysosome are membrane bonded sacs with digestive enzymes. These enzymes are made by RDR. Okay, these enzymes are made by which this one is lysosome. Okay, inside the lysosome there are many digestive enzymes are present. Okay, so this enzyme are made by RDR. Okay, lysosome are a kind of waste disposal system of the cell. It helps to keep the cell clean, digesting and poison material as well as burn out cell organelles. It means it clean the cell. Okay, it digests all the waste material which is inside the cell. Poison material entering the cell such as bacteria or food as well as old organelles end up the lysosome, which break complex substance into a simpler substance. Lysosomes are able to do this because they contain powerful digestive enzymes capable of breaking down all organic material. It means if the any cells organelles are dead, lysosome also eat them, like because in the lysosome there is a many very powerful digestive enzymes are present. During the disturbance in the cellular metabolism, for example, when the cell gets damaged, lysosome may burst and the enzyme digest their own cell. Therefore, lysosome are also known as suicide bags of a cell. It means if any cells are in danger, okay, in the danger, then the lysosome burst, okay, and then the all organelles inside the cell, just because of because the lysosome have the powerful digestive enzymes, that's why when the lysosome bursts, the all cell organelles are what? Finished. Okay, so that's why it is also called suicide bag. So this is all about. Okay, let's read about mitochondria also. Mitochondria are known as powerhouse of the cell. Mitochondria have a two membrane covering. Okay, mitochondria have two membrane. Okay, first one is outer and second this one is inner. And inside matrix, the outer membrane is porous while the inner membrane is deeply folded. Increased surface area ATP. Okay, this fold is called, and if we zoom out on the fold, it contains this type of called, this one is oxysome. Okay, this helps into ATP production. Generating chemical reaction, the energy required for a various chemical activities needed for life is released by the mitochondria in form of ATP, adenosine triphosphate molecule. Okay, I have already told that it helps in ATP production. The body uses energy stored ATP making new chemical compound and for mechanical work. Okay, mitochondria are a strange organelle in the sense that they have own. It is a strange organelle because they contain their own DNA, own ribosome, and also only two types of things they contain own. Therefore, mitochondria are able to make some of their own protein. Okay, that's why it can able to make the own protein. Okay, that's why it is called the powerhouse of the cell. Okay, and the lysosome is also most powerful, that it have the most powerful digestive enzyme to digest the wastage of the cell. Okay, next one is plastid vacuoles, and after that this one is cell division. This one is the process of cell division. Okay, so only 3 topics are left. So in next video we will complete this chapter, and after completing this chapter we will discuss some question which is inside the book. Okay. there are 10 questions are available, so we will read about the questions. Okay, that's all. Thank you.